I'm Zoltan Arany. I'm the chair of the Cell Biology, Physiology, and Metabolism program at Penn, and I want to tell you a little bit more about our program. Let me switch to the slides, and um, and let me tell you a little bit more about our program. So we are a subgroup of CAMB, and we're focused on, well, cell biology, physiology, and metabolism. So why those three together? Because they're intricately related. They meld form, function, and the means to generate and regulate life, basically. And why do I have parentheses here around physiology and metabolism? Because it's not just cell physiology and metabolism we're interested in, it's the entire program. It's the entire organism, ranging from yeast to flies to humans even. All of it is covered in our program. There are a lot of highlights to the CPM program, but let me just give you four of them. We use and develop state-of-the-art innovative imaging approaches to image often at the single molecular level, like here, cellular biology in action. This is a beautiful images of the cardiomyocyte from the Penn lab. Our program prides itself on rigorous quantitative analysis, whether it's in imaging or in metabolic flux analyses or whatever. We believe that careful quantitative analysis gives you true mechanistic insight, whereas superficial observations can often lead you wrong. We're also a highly collaborative group. You'll hear this many times at Penn, but I think this is the best part of this place, is that people really do love working together and use multidisciplinary approaches to attack big questions. And finally, we also pride ourselves in doing fundamental basic research, but that is also often with clear medical relevance. And we leverage the unique access to what is an incredibly rich academic medical center, and indeed one of the best in the world. So as part actually of this effort of Penn to integrate departments and basic research and clinical relevance, they've developed a number of institutes that span across departments. And CPM is intimately involved with three of them: the Penn Cardiovascular Institute, the Penn Muscle Institute, and the Institute of Diabetes, Obesity, and Metabolism. And you're going to see numerous times over the next few slides, these logos appear because our faculty are closely associated with all three of these. And again, this reflects our keen interest in melding basic cell biology and physiology with clinically relevant questions and leveraging the power of being associated with a preeminent clinical center. So let me tell you a little bit about some of the labs very quickly. I'll start with the cell biology part component. Sorry, I'm jumping slides here. I'll start with the cell biology component. This is going to go real fast, so don't sweat the details. You can always come back and look at each slide. This is sort of a 20,000-foot view of what's going on in the lab. So this is Kevin Foskett's lab. Kevin is also the chair of the physiology department, and he's interested in the biophysics and physiology of endoplasmic reticulum to mitochondrial calcium signaling and ion channels. His lab has made really seminal discoveries in the mechanism of calcium handling and implications, for example, in Alzheimer's disease. And most recently, his lab has actually been at the forefront of understanding how calcium enters mitochondria in the wake of the recent discovery of the calcium mitochondrial uniporter. Mark Kahn is a cardiologist in the Cardiovascular Institute. He's a world-renowned vascular biologist. He studies multiple aspects of vascular biology, each of them with very important clinical relevance, including, for example, how vessels prevent blood clots from forming while you're on an airplane, or so some people are born with vascular malformations, or how lymphatics form, which are key for draining fluid, obviously, including diseases such as, for example, heart failure. Vicky Marks studies the fundamental cell biology of a very specific cell type organelle called the lysosomal related organelle, LRO. Melanosomes are an example of that, the pigment-producing organelles in the skin and the eye. And there are diseases where these organelles don't function properly, including some forms of albinism. And the Marks lab is trying to understand why these things don't work, while at the same time studying fundamental biology of lysosome and LROs. Milike Lakadamiali is developing really super cool resolution microscopy, which her lab then uses to study the spatial temporal organization of the cell at the near molecular level. So, for example, live imaging of organelle transport along intermediate filaments, or looking at chromatin structure visually with microscopy. These are very cool approaches. The Good Lab is asking some really basic cell biology questions, verging on philosophical questions like why are cells the size that they are? His lab develops tools, including optogenetics, to look at cytoskeletal patterning and even synthesizing new cells to see how size and shape of cells affect fundamental processes like genome activation, for example. So those are just a few examples. There's very strong interconnections between, as I said, between cell physiology, metabolism, and biology. And so, I'm splitting these into three groups, but really they're intimately interconnected. So let me give you some physiology examples. Ken Margulies, who's also an active cardiologist, and he's the fellowship director of the heart failure and transplant program, actually. He's been collecting human hearts post-transplant for many years, and he's developed unique methodologies to isolate cardiac cells from these hearts, as well as using iPS cells to generate specialized microtissues that more truly mimic the true cardiac microenvironments. And using these techniques to ask fundamental questions about cardiac mechanobiology, especially in the context of cardiotoxicity and allograft rejection. The Prosser lab, Ben Prosser, works very closely with Ken, actually, whom I just told you about, and they've had some spectacular recent discoveries uncovering new roles of microtubules in the cytoskeleton and in the mechanobiology of the cardiomyocytes, with some potentially actually imminently translatable implications in clinical heart failure. These are really very exciting studies. In addition to that, Ben Prosser's lab has also developed a new direction recently after his daughter, Lucy, this is a very cute video of his daughter, was diagnosed with a rare genetic neurodevelopmental disorder. Ben has been working with groups at CHOP to develop, CHOP being the Children's Hospital of Philadelphia next door, to develop novel therapeutic approaches to correct these disorders, and these are actually now already being pursued through partnership with pharmaceuticals. The Ostap lab, Mike Ostap, who's also the director of the Penn Muscle Institute, looks to understand the cellular machinery responsible for powering cell movement and shaping the architecture of cells, tissues, and organs. So we're talking here about very fundamental stuff, molecular motors, that, for example, powers cell migration, muscle contraction, and the transport of internal cell compartments, for example. Yale Goldman, who used to be the director of the Penn Muscle Institute before Mike Ostap, is, like Mike, also interested in understanding molecular motors, and in particular relating their structural changes to the enzymatic reactions and the mechanical steps of the energy transduction. So again, very fundamental and quantitative physiology and cell biology. So that involves a lot of metabolism, and a lot of us are focused on metabolism itself, whether it be in the cell or the organism. And let me give you a couple of examples of that. I'll start with my own lab. We love everything metabolism. I too am a practicing cardiologist, so in particular we focus on cardiovascular metabolism on three fronts: metabolism in blood vessels, metabolism in the heart, and also integrated organismal metabolism. We use all sorts of tools, ranging from isotopic metabolomics in mice to CRISPR screens to human genetics and studies, all in an effort to understand the role of metabolism in disease, especially in heart failure, diabetes, and metabolic syndrome. The Bauer lab, one floor upstairs from us, studies aging and metabolism. They use similar mouse and metabolomic studies as we do. A lot of Joe's work has been on mTOR signaling and on NAD biology and their relationship to aging. Both of these are hot topics these days. The Tichenal lab, Paul Tichenal, also studies mTOR, like Joe, but he looks at it in the context of liver biology, and especially in the context of non-alcoholic fatty liver disease, which is hugely relevant these days, probably affects about a third of the US population. Back down to our floor, Dan Kelly, who is also the director of the Cardiovascular Institute, probes deeply into the transcriptional networks that control metabolism in the heart. Especially his lab has demonstrated recently the potential of ketone bodies to benefit heart failure, which is currently really a hot arena with a potential translation into the clinic. Li Ming Pei, who's a CHOP at Children's Hospital of Philadelphia, but also part of our program, like Dan, also looks at transcriptional networks that regulate metabolic programming in the heart. And he's spearheaded some very sophisticated single-cell and single-nuclei transcriptomics in the heart, which he's leveraging to make really interesting new discoveries on that front. The Amita Segal lab, Amita is a Howard Hughes investigator. She seeks to understand the molecular and cellular networks that drive rhythmic behavior, like sleep. Her lab works largely with the fruit fly, but also translates findings to mammalian models. As I said, we're interested in not just the cell, but entire organs, entire organisms, including, of course, humans and human disease. And finally, but not least, Josh Dunayev studies iron handling. Iron is fundamental to metabolism, as you probably know. And he has a special focus on the retina, where it turns out iron plays a really important role. He's an international leader in this topic, and his studies are clearly indicating that mishandling of iron may actually well be the #1 cause of blindness in the United States. So there you have it, really, in a quick nutshell. CPM, we do a lot of really exciting stuff. We meet every other week for a free lunch, always really good food, for work in progress and really just sort of catch up with each other. It's really quite fun. We also have special seminars of student-invited speakers. And then most of our students, really our faculty as well, leverage our close relationship with the 3 institutes and attend each of their weekly seminar series that includes absolutely world-class researchers from around the country and often outside the country. And finally, we do take advising very seriously. I personally advise all the students for coursework, choice of labs, and whatever issue might come up. And we also have advisory teams. We take that very seriously. So the bottom line is that CPM is exciting. The labs have in common interest really a passion for mechanistic cell biology and quantitative physiology and metabolism. We have a drive to understand the functioning of cells in context, both in physiology and in pathology, and especially a strong connection between the basic research and human medicine, leveraging this access we have to what is a really spectacular world-class medical center. So I'll stop there. This is our website. This is my contact information. Please feel free to contact me at any time. And really, you should always feel free to call any PI in CPM or really anywhere else for that matter. And again, welcome to Penn.